Beneath your feet, right now, there is a world you have never been told about. Not metaphorically, not as a figure of speech. Literally beneath the ground you are standing on, beneath the asphalt, beneath the bedrock, beneath the continental crust that has supported every civilization in human history. There are living organisms. Organisms that eat rock. Organisms that breathe sulfur. Organisms that have been isolated from the surface of this planet for longer than the dinosaurs existed. And they are alive. This is not science fiction. This is microbiology, geology, and astrobiology working in concert to reveal one of the most disorienting discoveries of the last 50 years. Earth's deep subsurface is a biosphere. A second biosphere. One that exists entirely independently of sunlight, of photosynthesis, of the food chains and ecological systems that every school textbook taught you were the foundation of all life. The numbers shatter intuition. Scientists estimate that the total mass of microbial life living in the deep subsurface of Earth, in pores within rock, in fluid-filled fractures kilometers underground, in sediment layers compressed under pressures the human body could not survive for a single second, may be comparable to or even exceed the total biomass of all life on the surface. Every tree, every animal, every insect, every blade of grass, every coral reef, every human being who has ever lived. That entire system of surface life may be outweighed by organisms that have never once encountered daylight. Read that again. Let it settle. The surface world, the forests, the oceans, the savannas, the teeming, interconnected web of surface ecosystems that we have spent centuries studying and cataloging, may represent only a fraction of the living mass of this planet. The majority of Earth's biology could be underground, invisible, silent, and ancient beyond comprehension. How is that possible? How did we miss it for so long? The answer is not negligence. It is physics. The crust of the Earth is not empty space. It is not a hollow chamber a scientist can crawl into with a flashlight. It is solid rock, granite, basalt, shale, limestone, compressed under pressures that increase by roughly 1 atmosphere for every 10 meters of depth. Drill 1 kilometer down, and the surrounding rock exerts a pressure equivalent to 100 atmospheres. Drill 10 kilometers, and that number climbs to approximately 1,000 atmospheres, a pressure that would crush most mechanical equipment designed for surface conditions. The deep Earth is physically hostile to human instruments, to human bodies, and to nearly every form of technology we routinely use to explore the world. And yet, life is there. The first serious scientific evidence began to accumulate not in a laboratory, but in an American gold mine in the 1980s. Researchers from the University of Georgia, drilling into savanna river site sediments in South Carolina at depths ranging from 200 to 500 meters, began retrieving rock cores containing something no one expected: bacterial cells. Not contamination from the drilling process, the scientists had rigorous contamination controls, but genuine indigenous microbial communities living in rock formations hundreds of meters below the surface. The paper, published in Science in 1987 by Frank Waber and colleagues, detonated a quiet revolution in biology that has been building ever since. What made the discovery genuinely astonishing was not just the presence of life, it was the metabolic strategy. These organisms were not using photosynthesis. There is no sunlight at 500 meters depth. They were chemolithotrophs, organisms that derive energy directly from chemical reactions involving inorganic compounds. Some oxidize iron, some reduce sulfate into hydrogen sulfide, some consume hydrogen gas produced by a process called serpentinization, in which water reacts with iron and magnesium-rich rocks at depth to produce hydrogen as a byproduct. These organisms had built an entire metabolic economy around chemistry that the surface world barely touches. Serpentinization deserves a moment of attention because it is one of the most consequential geological processes for the question of life. When water infiltrates certain types of oceanic crust, specifically ultramafic rocks like peridotite, which are rich in olivine, a chemical reaction begins. The water oxidizes the iron in the rock, converting olivine to serpentine minerals. In doing so, it releases molecular hydrogen. That hydrogen, a potent energy source, drifts upward through rock fractures. For a chemolithotrophic microorganism living in those fractures, it is fuel. It is the equivalent of sunlight for a plant, except it requires no star, no surface, no sky. Serpentinization happens in the dark, under pressure, driven purely by the thermodynamics of mineral chemistry, and it has been happening on Earth for billions of years. The implications of that timeline are staggering. Life on Earth's surface is constrained in certain ways. It requires liquid water, yes, but it also depends on a star that is roughly 4.5 billion years old, in a specific phase of its life cycle, at a specific distance that maintains surface temperatures in a liquid water range. Remove the sun, dim it, move it, age it past the main sequence, and surface life collapses within geological eye blink. Deep subsurface life is not similarly constrained. It does not care about the luminosity of the sun. It does not require seasons or atmospheric oxygen or even a functioning hydrological cycle at the surface. Its energy source is the chemistry of the rock itself, driven by the residual heat of the planet's formation and the slow, grinding interaction of minerals and fluids. Some of the microorganisms retrieved from deep boreholes have generation times, the time it takes for a single cell to divide into two, measured not in minutes or hours, but in centuries, possibly millennia. The metabolic rates are so low that by surface standards, these organisms barely qualify as active. They are not dormant. Dormancy is a distinct biological state involving spore formation or suspended animation. These cells are metabolically active. They are eating. They are reproducing. They are, in the technical biological sense, alive. They simply do it at a pace that makes a glacier look hurried. Think about what that means for their age. A colony of bacteria living in a rock fracture 2 kilometers below the surface of South Africa, metabolizing at a rate where one generation takes 10,000 years. That colony may have been genetically continuous, parent to offspring, since before the last glacial maximum, since before the extinction of Neanderthals, since before any member of our species painted a hand on a cave wall. The lineage may predate human language, human tools, human cognition, and it has been there, in the dark, in the rock, the entire time. This is the world beneath the world. Scientists have now retrieved living microorganisms from depths exceeding 5 kilometers below the surface. The deepest confirmed biological samples come from gold mines in South Africa, specifically from the Witwatersrand Basin, where boreholes have penetrated ancient Precambrian rock formations. In fissures within that rock, researchers found communities of bacteria and archaea that had been isolated from the surface for an estimated 3 million to 12 million years. They were not fossils. They were not chemical traces. They were living cells, retrieved from a world that had been sealed off from the surface since before the earliest members of the genus Homo walked the African savanna. 12 million years of isolation. To calibrate that number against human experience, recorded human history spans roughly 5,000 years. The entirety of Homo sapiens as a species covers approximately 300,000 years. 12 million years ago, our evolutionary lineage had not yet diverged from the great apes. The organisms living in those South African rock fractures have been down there, unchanged, since a time when no human, no proto-human, and no ancestor we would recognize was alive anywhere on Earth. The scientific term for these organisms, broadly, for life that exists in extreme conditions previously considered incompatible with biology, is extremophiles. Within that category, organisms specifically adapted to life underground, in rock, are called endoliths. The name comes from Greek, endon, meaning within, and lithos, meaning rock. Within rock organisms. The classification encompasses multiple domains of life: bacteria, archaea, and in some cases even fungi and single-celled eukaryotes have been found at surprising depths. The existence of endoliths did not merely expand the boundaries of biology, it fundamentally revised the definition of a habitable environment. Before the discovery of deep subsurface life, the requirements for habitability were understood, intuitively if not always explicitly, to include proximity to an energy source, meaning a star. The entire framework of planetary habitability, from the early work of astronomers assessing the potential for life on other worlds, was built around the concept of a habitable zone, the orbital distance from a star at which liquid water could exist on a planetary surface. Too close, and the surface water boils away. Too far, and it freezes. The zone in between, sometimes called the Goldilocks zone in popular science writing, though that term is a significant oversimplification, was treated as the essential prerequisite for life. Deep subsurface biology did not merely challenge that framework, it demolished the assumption underneath it. If life can thrive at 5 kilometers depth, fueled entirely by rock chemistry, with no connection to the surface and no dependence on stellar radiation, then the habitable zone of a star is not the boundary of habitability. It is merely the boundary of surface habitability. The interior of a rocky planet, warmed by residual formation heat and radiogenic decay, laced with fracture systems carrying fluid, rich in reactive minerals, may sustain life regardless of its distance from its star. That realization has a trajectory. It points somewhere specific, and that somewhere is not far from here. The Witwatersrand Basin in South Africa is the largest known gold deposit on Earth. It stretches across an area of roughly 300 kilometers, and its deepest mines descend more than 4 kilometers below the surface, making them the deepest operational mines in human history. The Tautona mine, outside Carletonville, reaches a depth of 3.9 kilometers. At that depth, the ambient rock temperature climbs to 60 degrees Celsius. Ventilation systems run continuously just to keep the air breathable. The pressure on the human body at that depth is not immediately lethal, but it is relentless. A physical reminder that the deep Earth was not engineered for biological visitors. In 2006, a team of geoscientists and microbiologists led by Barbara Sherwood-Lawler of the University of Toronto and Tullis Onstad of Princeton University was collecting fluid samples from fractures in the ancient rock walls of Tautona. They were not looking for life in the way a biologist might scan a petri dish. They were performing isotopic analysis, measuring the chemical signatures of the water itself, looking at the ratios of different isotopes of hydrogen, oxygen, and noble gases like helium and neon to determine how long that water had been isolated from the surface hydrological cycle. The technique is precise. Radioactive isotopes decay at known rates, and the accumulation of certain daughter isotopes in an isolated fluid acts as a clock. The older the water, the more the isotopic signature drifts from surface values. What they found in those fractures stopped the analysis cold. The water was old. Not geologically old in the casual sense scientists sometimes use when discussing sedimentary formations. The isotopic signatures indicated that some of the fluid samples had been isolated from the surface for between 1.5 billion and 2.6 billion years. That number requires repetition. 2.6 billion years. The water trapped in those rock fractures had last been in contact with the surface atmosphere during the Archean Eon, a period of Earth history so remote that the atmosphere itself was fundamentally different, with negligible free oxygen, dominated by nitrogen and methane and carbon dioxide. The continents as we know them did not yet exist. The moon was closer to Earth. The oceans were warmer, possibly tinged with dissolved iron that gave some ancient seas a greenish hue, and a pocket of water seeping into a fracture in the growing crust was sealed inside. It stayed there. For 2.5 billion years, that water did not move. It did not evaporate. It did not rejoin the water cycle. It sat in the dark, under pressure, in rock, while the entire history of complex life on Earth unfolded above it. While the Cambrian explosion populated the oceans with the ancestors of every animal alive today. While the first fish developed jaws. While plants colonized the land. While dinosaurs rose, dominated the planet for 165 million years, and were erased by an asteroid strike. While the first mammals grew larger. While the first primates climbed into trees. While the first hominins walked upright across an African plain. All of that, the entire cinematic sweep of life's history on this planet's surface, occurred while that water remained sealed, motionless, in the rock below. And in that water, isolated, ancient, chemically altered by centuries upon centuries of reaction with the surrounding minerals, there were microorganisms. Onstad's team, working in collaboration with microbiologists including Li Hung Lin, had already reported in 2006 in the journal Science the discovery of a sulfate-reducing bacterium living in fracture water retrieved from 2.8 kilometers depth in the same basin. The organism, belonging to a group related to the genus Candidatus desulforudus audax viator, a name drawn with fitting drama from Jules Verne's Journey to the Center of the Earth, was found to be the sole detectable inhabitant of its isolated ecosystem. A single species, alone, completely self-sufficient. It fixed its own nitrogen, synthesized its own organic carbon compounds, and derived energy from the chemical reduction of sulfate using hydrogen produced by the radiolysis of water, the splitting of water molecules by the radioactive decay of uranium, thorium, and potassium in the surrounding rock. That energy chain deserves careful attention because it severs the last conceptual thread connecting deep life to the surface. Radiolysis does not require the sun. It does not require surface geology to be active. It does not even require the planet to be geologically young. As long as radioactive isotopes are present in the rock, and they are present in virtually all crustal rock in varying concentrations, water molecules will be split, hydrogen will be produced, and an organism capable of using that hydrogen as an electron donor in a sulfate reduction reaction will have fuel. The energy source is nuclear, ancient nuclear. The radioactive elements in the Witwatersrand rock are primordial. They were synthesized in stellar explosions before the solar system formed and incorporated into the Earth during planetary accretion 4.5 billion years ago. Desulforudus audax viator is, in a very literal sense, running on energy derived from the deaths of stars that predate our sun. The isolation of this organism raised a question that divided researchers for years. Was it genuinely a complete, self-contained ecosystem? Every surface ecosystem on Earth, however simple, is ultimately entangled with others through atmospheric exchange, water flow, the movement of organisms. The concept of a single-species biosphere entirely closed, with no external biological inputs and no exports, had no precedent in surface ecology. It seemed almost theoretically impossible. Ecosystems, as traditionally understood, require diversity: producers, consumers, decomposers, recyclers. A single organism, no matter how metabolically versatile, seemed insufficient to close all the biogeochemical loops necessary for indefinite survival. But Desulforudus audax viator appeared to close them all. Its genome, sequenced in full by Onstadt and colleagues, revealed a remarkable degree of metabolic completeness. It carried genes for nitrogen fixation, converting inert atmospheric nitrogen dissolved in the fracture water into biologically usable ammonia. It carried genes for carbon fixation through the reductive TCA cycle. It carried genes for synthesizing all 20 standard amino acids, all nucleotide bases, all membrane lipids. Everything a living cell requires to build itself from scratch was encoded in a single genome, operating in a single organism, in a single fracture, kilometers underground. The genome also carried genes for sporulation, the ability to form dormant, highly resistant spores capable of surviving extreme conditions for extended periods. This capacity for dormancy is not merely a biological curiosity. It is a survival strategy of profound durability. Bacterial spores retrieved from amber have been revived after estimates ranging from 25 million to 40 million years of dormancy, though those figures remain scientifically contested. What is not contested is that spores can survive conditions, heat, desiccation, radiation, pressure, that would destroy any vegetative cell. An organism that can both actively metabolize in fracture water for geological timescales and retreat into dormancy when conditions deteriorate is not merely surviving. It is playing a game with time on terms that make the human lifespan vanishingly brief. The discovery of Desulforudus audax viator reframed the entire scientific conversation about subsurface life. Before it, the deep biosphere was understood as a community, diverse microbial assemblages, impoverished compared to surface soils, but still composed of multiple interacting species. After it, the possibility of a genuinely isolated single-species, self-sustaining biosphere became real. And if it was real once, in one fracture, in one mine, in one geological province of one planet, the statistical implications were immediate and unavoidable. Those implications extended outward, They extended to Mars, whose subsurface has been shaped by similar Precambrian-era geological processes, and where liquid water likely persisted underground long after the surface became uninhabitable. They extended to Enceladus, Saturn's moon, where the Cassini spacecraft detected active water vapor plumes erupting from the South Pole. Plumes containing molecular hydrogen, silica nanoparticles consistent with high-temperature water-rock interaction, and organic compounds. They extended to Europa, where a liquid ocean believed to contain more water than all of Earth's oceans combined sites beneath an ice shell kilometers thick, in contact with a rocky seafloor where hydrothermal chemistry similar to Earth's deep subsurface almost certainly operates. The fractures in the Witwatersrand rock had pointed a searchlight at the outer solar system. But there was a problem. A fundamental, unresolved, deeply uncomfortable problem that the discovery of subsurface life created for astrobiology, for planetary science, and ultimately for the way scientists think about the boundaries of life itself. If life can persist in total isolation, fueled by nuclear decay, for timescales measured in billions of years, how do we detect it? The organisms found in Tautona and in dozens of subsequent deep borehole projects around the world are not accessible by telescope. They emit no electromagnetic signature that a spacecraft in orbit could detect. They produce no atmospheric biosignature in any quantity sufficient to be measured remotely. They are chemically subtle, biologically sparse by surface standards, and physically located beneath kilometers of solid rock that no current or near-future planetary probe can penetrate. The deep biosphere, in short, is scientifically transformative and observationally invisible. It changes everything we know about where life can exist, and it hides everything we would need to confirm that it does. That paradox is not merely frustrating, it is the central challenge of 21st century astrobiology, and it begins, not among the stars, but in the ancient pressurized dark beneath our own feet. The answer to how science is attempting to bridge that gap, how researchers are building the tools, the methods, and the theoretical frameworks to detect what cannot be seen, begins with understanding exactly how life survives under conditions that should, by every prior assumption, be lethal. Pressure is the first thing that kills surface biology underground. Not gradually, not with warning. Pressure at depth is immediate, structural, and absolute. The human body can survive, with technological assistance, pressures of perhaps 6 to 8 atmospheres before the physiological consequences become unmanageable. Nitrogen narcosis begins affecting cognitive function at depths equivalent to roughly 4 atmospheres in diving contexts. At 10 atmospheres, unprotected tissue begins to fail. At 100 atmospheres, the pressure at 1 kilometer depth in solid rock, no multicellular organism survives without extraordinary physical protection. At 1,000 atmospheres, the pressure at 10 kilometers depth, the molecular structure of most proteins begins to denature. Enzymes fold incorrectly. Cell membranes lose their selective permeability. The machinery of life, built for the gentle pressure regime of the surface, simply stops working. And yet the microorganisms retrieved from kilometers of depth are not merely tolerating pressure, they are exploiting it. Piezophiles, organisms specifically adapted to high-pressure environments, have evolved membrane compositions that remain fluid and functional at pressures that would rigidify and rupture standard biological membranes. The key is lipid chemistry. The cell membrane of any living organism is a lipid bilayer. 2 sheets of fat molecules arranged so that their water-repelling tails face inward and their water-attracting heads face outward, forming a flexible barrier that separates the cell's interior from the external environment. At surface pressure and temperature, this membrane maintains the precise degree of fluidity necessary for protein function, ion transport, and cellular signaling. Too rigid, and the proteins embedded in the membrane cannot rotate or flex as they perform their functions. Too fluid, and the membrane loses its integrity, and the cell's contents spill out. Temperature and pressure both affect membrane fluidity in the same direction. Cold makes membranes more rigid. High pressure makes membranes more rigid. An organism living at 4 kilometers depth at 60 degrees Celsius is dealing with 2 competing physical forces. The heat tends to fluidize its membranes, while the pressure tends to rigidify them. The result is a biochemical balancing act that evolution has been performing underground for billions of years. Piezophilic organisms adjust the ratio of saturated to unsaturated fatty acids in their membranes. Unsaturated fatty acids have kinked molecular chains that resist tight packing and maintain fluidity under pressure. Some deep subsurface archaea take this further, using a fundamentally different membrane architecture. Instead of the ester-linked fatty acid chains found in bacteria and eukaryotes, archaeal membranes use ether-linked isoprenoid chains that are intrinsically more resistant to both heat and pressure. This is one reason archaea, the third domain of life, phylogenetically distinct from both bacteria and eukaryotes, dominate many of the most extreme deep subsurface environments on Earth. Temperature presents its own gradient of challenge. The geothermal gradient, the rate at which temperature increases with depth into the Earth, averages roughly 25 to 30 degrees Celsius per kilometer of depth, though it varies significantly depending on local geology. Near tectonic boundaries, volcanic regions, and mid-ocean ridge systems, the gradient is steeper. In ancient tectonically stable cratons like the Kapval Craton underlying the Witwatersrand Basin, it is shallower. This gradient sets a hard physical ceiling on the depth at which any biology is currently known to persist. At approximately 120 degrees Celsius, a temperature that denatures most proteins and disrupts nucleic acid structure even under high pressure, life as currently characterized becomes thermodynamically untenable. That temperature is reached at different depths in different geological settings, roughly 4 kilometers in a geothermally active region, potentially 10 kilometers or more in a cold, ancient craton. The upper bound of 120 degrees Celsius is not a theoretical limit derived from first principles. It is empirical. It is the highest temperature at which living, reproducing organisms have been confirmed. The current record holders are hyperthermophilic archaea, primarily from hydrothermal vent environments, including Methanopyrus candeleri, which was shown to reproduce at 122 degrees Celsius under high-pressure conditions in laboratory experiments by Qasim Kashefi and Derek Lovely, published in Science in 2003. That organism does not live deep in the subsurface. It was isolated from hydrothermal vent deposits on the seafloor. But its biochemistry illuminates the same principles that allow deep subsurface thermophiles to function at elevated temperatures. Its proteins are stabilized by an unusually high density of ionic interactions and disulfide bonds. Its DNA is protected by unique histone-like proteins and by high intracellular concentrations of potassium and other solutes that suppress DNA denaturation. Its ribosomes, the molecular machines that translate genetic code into proteins, are reinforced with additional protein subunits that prevent thermal disassembly. Water availability underground is a third physical constraint, and it operates in ways that are less intuitive than pressure or temperature. In the deep subsurface, water is not absent. It exists in fractures, in pore spaces between mineral grains, and as chemically bound water within hydrated minerals. But its chemical activity, the thermodynamic measure of how available it actually is to participate in biological reactions, can be severely reduced by dissolved salts. Deep fracture waters are frequently hypersaline, with salt concentrations far exceeding that of seawater. Seawater contains approximately 35 grams of dissolved salts per liter. Some deep subsurface brines exceed 300 grams per liter, nearly 10 times saltier than the ocean. At those concentrations, osmotic pressure across cell membranes becomes extreme. Water moves out of cells toward the more concentrated external solution, desiccating the cell from the outside even when surrounded by liquid. Halophiles, salt-tolerant organisms, counter this by accumulating compatible solutes inside their cells. These are small organic molecules, glycine betaine, ectoine, trehalose, various amino acids, that raise the cell's internal osmotic pressure without interfering with enzyme function. The strategy is metabolically costly. Synthesizing compatible solutes requires energy and raw materials. In a low-energy, deep subsurface environment, where every calorie of chemical fuel is precious, the overhead of osmoprotection represents a significant fraction of the organism's total energy budget. This is part of why metabolic rates in deep subsurface organisms are so extraordinarily low. They are not merely slow because the chemistry is slow. They are slow because the organisms are rationing every unit of energy across multiple simultaneous survival demands: membrane maintenance, DNA repair, osmoprotection, and the minimal metabolic housekeeping necessary to avoid cellular disintegration. DNA repair is a non-negotiable energy expenditure at depth. The rock surrounding deep subsurface organisms is radioactive. Uranium, thorium, and potassium-40 decay continuously, emitting alpha particles, beta particles, and gamma radiation into the surrounding fluid and rock. While the radiation levels are low compared to, say, the environment immediately surrounding a nuclear reactor, the cumulative dose over geological timescales is enormous. A cell living in a uranium-bearing rock fracture for millions of years receives a total radiation dose that would, delivered rapidly at the surface, be instantly lethal to any surface organism. Deep subsurface organisms survive this not by being radiation-proof in some absolute sense, but by having exceptionally efficient DNA repair machinery that continuously corrects the damage. Deinococcus radiodurans, known from surface environments as the most radiation-resistant organism characterized, can reassemble its genome from hundreds of fragments after radiation-induced double-strand DNA breaks. Related repair mechanisms, though not always at the same extraordinary efficiency, operate in deep subsurface communities, allowing them to sustain viability under chronic low-level irradiation for durations that have no parallel in surface biology. The energy flows sustaining all of this biological activity underground are almost incomprehensibly small by surface standards. A square meter of sunlit grassland captures roughly 5 to 20 watts of solar energy through photosynthesis under ideal conditions. A comparable cross-section of deep subsurface rock at 2 kilometers depth receives a chemical energy flux from radiolysis and geochemical reactions on the order of nanowatts to microwatts per square meter. The difference spans roughly 10 orders of magnitude, 10 billion times less energy per unit area. That number reframes everything observed about deep subsurface biology. The extreme slowness of reproduction, the ultra-low biomass densities, the minimal metabolic rates. These are not signs of organisms barely clinging to existence. They are signs of perfect adaptation to an energy regime that the surface biosphere cannot access and has never needed to. Natural selection operating in the deep subsurface for billions of years has produced organisms tuned with extraordinary precision to extract maximum biological productivity from minimum chemical input. They waste nothing. They repair everything. They wait. The geochemical cycles that feed this hidden biosphere form closed loops of elegant efficiency. Sulfate-reducing bacteria consume hydrogen and sulfate, producing hydrogen sulfide as waste. Sulfur-oxidizing organisms, where they coexist, consume that hydrogen sulfide and regenerate sulfate. The sulfur cycles between oxidized and reduced forms, carrying energy around the loop, sustaining both metabolic strategies indefinitely as long as hydrogen from radiolysis or serpentinization continues to enter the system. Iron undergoes similar cycles. Ferric iron reduced to ferrous iron by iron-reducing bacteria, reoxidized abiotically or biotically, cycling through oxidation states and releasing energy at each transition. These are not simple reactions. They are planetary-scale biogeochemical engines running silently, continuously, in the dark, independent of the surface world's oxygen-driven metabolism. Carbon moves through these systems too, though slowly. Some deep subsurface organisms fix inorganic carbon, carbon dioxide dissolved in fracture water, into organic compounds through the reductive acetyl-CoA pathway, one of the most ancient carbon fixation pathways known, likely predating the Calvin cycle used by photosynthetic organisms by hundreds of millions of years. The organic carbon produced is consumed by other community members, and eventually the carbon is respired back to carbon dioxide, completing a cycle that has no connection to the atmospheric carbon cycle above. In geological terms, this represents a buried carbon reservoir of uncertain but non-trivial size, cycling on timescales of millions to hundreds of millions of years. The deep biosphere is not a footnote to Earth's carbon cycle. It is a parallel system running beneath it, operating at tempos and scales that conventional biogeochemical modeling had, until very recently, entirely failed to account for. And within this system, this ancient, pressurized, radioactively fueled, chemically closed biological underworld, there are signals. Chemical signatures, isotopic anomalies, biological markers preserved in rock formations that point towards something the data had not, until a specific set of experiments in the early 2000s been able to fully explain. Something in the deep rock is producing compounds that should not, by purely abiotic chemistry, be there in those concentrations. And tracking the source of those compounds led researchers to a discovery that fundamentally altered the scientific estimate of how much life the deep Earth actually contains. In 2008, an international consortium of scientists operating under the Deep Carbon Observatory, a decade-long research program funded primarily by the Alfred P. Sloan Foundation and involving more than 1,000 researchers across 50 countries, began the most systematic attempt in history to quantify the total mass of life living beneath Earth's surface. The project was not a single experiment. It was a coordinated global effort spanning ocean drilling programs, continental borehole sampling, mine-based microbiology, and geochemical surveys, pulling together data from sediment cores retrieved from the bottom of the Pacific, boreholes drilled into the Canadian Shield, fluid samples collected from mines in Finland, South Africa, and Canada, and rock cores from the Integrated Ocean Drilling Program's archives stretching back decades. The final synthesis, published in a series of papers culminating in a landmark 2018 summary in the American Geophysical Union Journal and presented at the AGU fall meeting that year, produced a number that restructured the entire field. The total mass of carbon locked in living organisms in Earth's deep subsurface, bacteria, archaea, and other microbial life existing below 15 to 20 meters depth, the conventional boundary below which solar energy can no longer be considered even an indirect metabolic driver, was estimated at between 15 and 23 billion tons of carbon. To calibrate that figure, the total carbon mass of all humans alive on Earth today is approximately 60 million tons. The deep biosphere outweighs humanity by a factor of roughly 300. It contains more carbon than all the world's forests combined. It represents somewhere between 70 and 80% of Earth's total microbial biomass, and between 15 and 20% of all living biomass on the planet by carbon mass. The surface world, forests, oceans, soils, all animal life, every ecosystem visible from space, accounts for the remaining fraction. This was not a refinement of previous estimates. It was a categorical revision. Earlier models, working from limited borehole data and extrapolating cautiously, had placed deep subsurface biomass at figures roughly an order of magnitude lower. The Deep Carbon Observatory's systematic sampling revealed that life was not merely present in isolated pockets below the surface. It was pervasive. It existed in virtually every geological setting sampled: oceanic sediments, continental crystalline basement rock, sub-seafloor basalt, deep aquifer systems, evaporate formations. The only consistent predictor of its absence was temperature exceeding the known biological threshold, and even at the margins of that threshold, organisms were found functioning at metabolic rates so slow they were nearly indistinguishable from abiotic chemical background noise. The oceanic component of the deep biosphere proved particularly surprising. Beneath the seafloor, beneath the sediment layers that accumulate on the ocean bottom over millions of years, lies basaltic oceanic crust, formed continuously at mid-ocean ridges and destroyed at subduction zones over cycles of roughly 200 million years. For most of the 20th century, this rock was considered essentially sterile. The temperatures near ridge systems were too high. Away from the ridges, the rock was cold but dense and seemingly impermeable. There was no obvious energy source. There was no reason to look. The first systematic evidence that oceanic crustal basalt harbored living communities came from work by Katrina Edwards at the University of Southern California and colleagues published in the early 2000s. Examining basalt samples recovered from ocean drilling program cores, they detected not just DNA, which could theoretically be a contamination artifact or a fossil trace, but intact cell membranes, intact metabolic enzyme activity, and isotopic signatures of active sulfur metabolism. The basalt was alive. And oceanic basalt is not a minor geological feature. It underlies the entire ocean floor, approximately 60% of Earth's surface. If microbial life is present throughout that rock at even the sparse densities detected in core samples, the total biomass implication is enormous. Subsequent work refined the picture. Circulation of seawater through the porous and fractured structure of young oceanic basalt a process called hydrothermal circulation that moves an estimated 5 times the total volume of Earth's oceans through the oceanic crust every million years, delivers chemical reactants continuously into the rock. Oxygen, sulfate, and organic compounds from the overlying water column percolate downward. Reduced minerals in the basalt react with the infiltrating fluid, releasing energy. Microbial communities positioned along these fluid pathways harvest that energy. The entire oceanic crust functions, in biogeochemical terms, as a slow-motion chemical reactor in which biology participates as both catalyst and product. The Deep Carbon Observatory results forced a parallel revision in how scientists interpreted existing geochemical data from sediment cores. For decades, anomalous methane concentrations in marine sediments had been attributed primarily to abiotic thermogenic processes, the breakdown of organic matter under heat and pressure, producing methane without biological involvement. Detailed isotopic analysis, particularly measurements of carbon-13 to carbon-12 ratios in sediment methane, revealed that a substantial fraction of that methane carried biological isotopic signatures. It was biogenic, produced by methanogenic archaea living in the sediment, consuming buried organic carbon and exhaling methane as a metabolic byproduct. These methanogens were living in sediments hundreds of meters below the seafloor, at temperatures and pressures and in geochemical conditions that had not previously been modeled as biologically productive. The methane they produced accumulated in enormous quantities. At certain depths and pressures, methane combines with water to form methane hydrates, ice-like crystalline structures in which methane molecules are trapped inside cages of water molecules. The global inventory of methane stored in seafloor hydrates is estimated at somewhere between 1 and 500 gigatons of carbon, though the uncertainty range remains wide. A significant fraction of that inventory was generated biologically by archaea living in the sediment above the hydrate stability zone, over millions of years of slow metabolic activity. The deep biosphere had been quietly filling a planetary-scale methane reservoir without anyone knowing the organisms responsible were there. That realization had implications beyond pure biology. Methane is a potent greenhouse gas, roughly 80 times more effective than carbon dioxide at trapping heat over a 20-year timescale. The stability of seafloor methane hydrates is sensitive to temperature and pressure. As ocean bottom water temperatures rise, hydrate stability decreases. The question of how much biologically produced methane might be mobilized from destabilizing hydrates under future climate scenarios became, overnight, a problem that required understanding the deep biosphere to model accurately. Life underground was not merely an academic curiosity, it was a variable in Earth's climate system that no prior climate model had included. The revised biomass estimates also forced a rethinking of the deep biosphere's role in the global carbon cycle over geological time. Surface carbon cycling, the exchange of carbon between the atmosphere, oceans, vegetation, and soils, operates on timescales of years to millennia. The deep biosphere cycles carbon on timescales of millions to hundreds of millions of years through burial, metabolism, and the slow return of carbon to the surface via geological uplift, volcanism, and fluid migration. These two carbon cycles are not independent. They are connected through the subduction of organic carbon into the mantle, the release of carbon-bearing fluids along subduction zones, and the weathering of ancient organic-rich rock formations uplifted by tectonics. The deep biosphere sits at the interface of biological and geological carbon cycling in a way that makes the distinction between the two nearly meaningless at long timescales. The scientific community's response to the Deep Carbon Observatory findings was not uniform acceptance. Biomass estimates for the deep subsurface carry substantial uncertainty. The sampling density, however dramatically improved over prior efforts, still covers a vanishingly small fraction of the total volume of rock involved. Extrapolating from borehole samples to global inventories requires assumptions about the spatial distribution of life that remain incompletely validated. Some researchers argued the estimates were too high, pointing to evidence that many DNA sequences recovered from deep samples represented dead cells or free DNA adsorbed onto mineral surfaces rather than living organisms. Others argued the estimates were conservative, noting that the deepest and most geologically remote environments remain almost entirely unsampled. What was not disputed was the direction of the revision. Every new dataset, every new borehole, every new analytical technique applied to deep subsurface samples had pushed the estimate of deep life's abundance upward, not downward. The trend line was unambiguous. The deep biosphere was larger, more widespread, and more metabolically diverse than any previous model had allowed. And embedded in the Deep Carbon Observatory dataset was a finding that had gone relatively underreported outside specialist circles, but that carried implications extending far beyond Earth. In multiple geographically separated sampling sites, oceanic basalt from the Atlantic, continental basement from Scandinavia, deep sediment from the Pacific, the consortium detected organisms from phylogenetic lineages with no known surface relatives. Not merely organisms adapted to extreme conditions from lineages that also contain surface-dwelling members. Organisms representing deep branches of the tree of life with no characterized counterparts anywhere in surface ecosystems. Lineages that appear, based on molecular clock estimates, to have diverged from their nearest known relatives hundreds of millions to over 1 billion years ago. The deep Earth had not merely preserved ancient life. It had incubated evolutionary trajectories entirely separate from those that produced the surface biosphere. While evolution proceeded on the surface, driving the diversification of animals, plants, fungi, and the vast majority of microbial life known to science, something else had been diverging slowly in the rock. Driven by different selective pressures, fueled by different energy sources, shaped by different geochemical environments, these deep lineages had been accumulating genetic and metabolic novelty in isolation for timescales that dwarf the entire history of complex surface life. What those lineages are capable of, biochemically, metabolically, evolutionarily, is not yet known. The tools to characterize them are still being developed. The genomic databases required to interpret their gene sequences are still being built. The culture techniques necessary to study their physiology in the laboratory are still being refined, because organisms adapted to 4 kilometers of pressure and 60 degrees of heat do not survive removal to a surface laboratory without extraordinary technical precautions. The answers, when they come, will not merely describe what lives underground. They will redefine what life is. 70% of all bacteria and archaea on Earth live underground. Not some, not a notable fraction. 70%. The surface world, every soil microbiome, every ocean water column, every hot spring, every forest floor, every human gut, contains less than 1/3 of Earth's total prokaryotic life by cell count. The deep biosphere, spanning a volume of approximately 2.3 billion cubic kilometers, nearly twice the volume of all the world's oceans combined, is not a marginal addendum to life on this planet. It is the dominant theater of microbial existence. That number, 2.3 billion cubic kilometers, requires a moment of reckoning. All of Earth's oceans together hold approximately 1.3 billion cubic kilometers of water. The habitable volume of the deep subsurface exceeds that by nearly 1 billion cubic kilometers. It extends from a few meters below the surface down to wherever temperature and pressure finally extinguish the last metabolic spark. Through sediment, through fractured basalt, through ancient crystalline basement rock, through the porous matrix of continental aquifer systems, through the deepest accessible reaches of mine shafts and scientific boreholes. It is the largest continuous habitable environment on Earth. And until the last decades of the 20th century, science did not know it existed. The genetic diversity embedded in this hidden volume is, if anything, more disorienting than its physical scale. When the Deep Carbon Observatory's Census of Deep Life, a coordinated sequencing effort that applied high-throughput environmental DNA analysis to hundreds of deep subsurface samples from around the world, compiled its results, researchers found that the number of distinct microbial lineages detected underground was comparable to, and in some analyses exceeded, the genetic diversity of the entire surface biosphere. Millions of distinct microbial types. The vast majority of them had no characterized representatives in any culture collection, any genomic database, any prior scientific literature. They were, in the precise technical sense of the term, unknown to science. The census team described this mass of uncharacterized deep life as microbial dark matter, a direct parallel to the cosmological concept, invoked not for rhetorical effect, but because the analogy is structurally accurate. Just as dark matter constitutes the majority of the universe's mass while remaining undetectable by direct electromagnetic observation, microbial dark matter constitutes the majority of Earth's biological diversity while remaining invisible to any detection method that requires cultivating organisms under surface conditions. Cultivation is the bottleneck. To study a microorganism in traditional microbiology, you grow it. You isolate it, feed it nutrients, incubate it, watch it reproduce, characterize its biochemistry. The entire edifice of classical microbiology, the identification of pathogens, the development of antibiotics, the mapping of metabolic pathways, was built on cultivation. But deep subsurface organisms cannot be cultivated by surface methods. Remove them from 4 kilometers of pressure, and they die. Remove them from their specific geochemical environment, their precise mixture of electron donors and acceptors, their particular mineral substrate, their exact temperature regime, and their metabolism collapses. Decades of attempts to culture deep subsurface organisms in surface laboratories produced almost nothing. The organisms simply refused to grow. The solution came from a methodological revolution that bypassed cultivation entirely. Metagenomics, the direct sequencing of all DNA present in an environmental sample without first isolating or growing any individual organism, allowed researchers to read the genetic content of a deep subsurface community directly from a rock core or fluid sample. No cultivation required. The DNA was extracted, fragmented, sequenced, and computationally assembled into partial or complete genomes representing every organism present in the sample. Applied to deep subsurface samples beginning in the early 2000s and accelerating dramatically through the 2010s as sequencing costs collapsed, metagenomics produced a portrait of deep life that cultivation had been unable to sketch. What the metagenomic portraits revealed was not merely diversity, it was metabolic novelty. Genes encoding enzymes for reactions with no known surface analog. Biochemical pathways assembled in configurations that surface metabolic models had not predicted. Some deep subsurface archaea carry genes for a form of carbon fixation that operates in conditions of near-zero energy flux, a metabolic strategy so parsimonious that it approaches the theoretical minimum energy required to maintain cellular organization. These organisms are not simply slow. They have fundamentally different metabolic architectures, tuned over billions of years of evolution under energy regimes that surface life has never experienced. Among the most striking metagenomic findings was the discovery, in multiple geographically separated deep subsurface environments, of organisms carrying gene cassettes for the anaerobic oxidation of methane coupled to sulfate reduction. A metabolic partnership previously known only from marine sediments, where it is carried out by consortia of archaea and bacteria working in tight physical association. In the deep continental subsurface, single organisms appeared to encode both sides of this partnership within a single genome. The metabolic cooperation that on the surface requires 2 distinct organisms had been, over geological time, internalized into one. This is not a minor biochemical curiosity. It represents a form of evolutionary integration, the compression of a multi-organism metabolic system into a single cellular unit, that has profound implications for understanding how complex metabolic pathways evolve and how the boundaries between organisms and ecosystems blur at the scale of deep geological time. The isotopic record preserved in ancient rocks adds a temporal dimension to this picture that direct biological sampling cannot provide. Carbon isotope ratios in carbonates and organic matter preserved in Archean sedimentary formations, rocks dating to 3.5 billion years ago, carry fractionation signatures consistent with microbial metabolism. The specific pattern of carbon-13 depletion in these ancient organic residues matches the fractionation produced by certain autotrophic carbon fixation pathways, including the reductive acetyl-CoA pathway used by many deep subsurface chemolithotrophs today. This is not proof that deep subsurface life existed 3.5 billion years ago. It is consistent evidence that the metabolic strategies now operating kilometers underground were among the earliest forms of biological carbon cycling on this planet. The implication reaches back to the origin of life itself. The hydrothermal vent hypothesis, first articulated systematically by geologist Michael Russell and chemist William Martin, proposes that life originated not at the surface, but in alkaline hydrothermal vent systems on the early seafloor, where natural proton gradients across inorganic mineral membranes provided the thermodynamic driving force for the first metabolic reactions. These systems are chemically and energetically similar to the serpentinization-driven environments that sustain deep subsurface life today. If life began in conditions resembling those now found kilometers underground, then the deep biosphere is not merely a peripheral curiosity. It may be the direct descendant of the original biological environment, the ecosystem most similar to the one in which the first self-replicating chemistry assembled itself into the first cells. Surface life, by this reading, is the derived condition. The forests, the oceans, the photosynthetic ecosystems that capture sunlight and drive the atmospheric and hydrological cycles we think of as the foundation of the living world. These are evolutionary elaborations built on top of a deeper, older, more fundamental biology. The oxygenation of the atmosphere approximately 2.4 billion years ago, driven by the proliferation of photosynthetic cyanobacteria, was catastrophic for many of the anaerobic organisms that had dominated surface environments before it. The organisms that survived the Great Oxidation Event intact, in large numbers, were those already living below the reach of the newly toxic oxygen-rich atmosphere. The deep subsurface communities, sealed in their fractures, indifferent to the chemical transformation occurring kilometers above them. They endured. They are enduring still. The 4 global microbial populations identified in a 2025 study published in ISME Communications, analyzing deep terrestrial subsurface groundwaters across 4 continents, showed consistent metabolic strategies and genetic adaptations that transcend geography. The same core lineages, carrying the same functional gene repertoires, appear in deep aquifer systems in Finland, South Africa, Canada, and Japan. These populations are not being transported between sites by any surface mechanism. Their geographic ubiquity reflects ancient dispersal through the deep crustal fluid systems of a younger Earth, followed by billions of years of in situ evolution and isolation. They are globally distributed relics of an early biosphere, preserved in the rock like biological amber, still metabolizing, still reproducing, still present. Every borehole drilled into ancient crystalline basement rock around the world now finds them. Every deep mine that pumps fluid from fractures finds their chemical signatures: anomalous hydrogen sulfide concentrations, isotopically light carbon in dissolved inorganic carbon pools, methane with biogenic isotopic fingerprints. The evidence is not subtle once you know what to look for. It is in the chemistry of the water, in the isotopic ratios of the gases, in the molecular weight distributions of the dissolved organic compounds, in the electrical signatures of microbial biofilms detectable by downhole geophysical instruments. The subsurface is not quiet. It is active, chemically complex, and biologically saturated at densities that prior science had no framework to anticipate. And the full extent of that activity, the total depth, the total volume, the total diversity, remains unknown. The deepest confirmed life sits at approximately 5 kilometers below the continental surface. The temperature limit suggests biology could persist to 10 or 12 kilometers in cold ancient cratons. Between 5 and 12 kilometers lies an unexplored volume of rock larger than the entire explored biosphere, unsampled, unsequenced, biochemically invisible to current technology. Whatever lives there has been living there, in the dark, under pressure, on the energy of decaying atoms, since before the first animal drew breath. There is a borehole in northwestern Finland, drilled into Precambrian bedrock that formed approximately 1.8 billion years ago, that has been continuously monitored for microbial activity since the early 2000s. The rock it penetrates is among the most ancient and geologically stable on Earth, part of the Fennoscandian Shield, a craton that has not been substantially deformed by tectonic activity since the Proterozoic. The water that seeps from fractures in that rock is anoxic, saline, and chemically reducing. It carries dissolved hydrogen, methane, and hydrogen sulfide. Its isotopic composition indicates isolation from the surface hydrological cycle for millions of years. And it contains living archaea, metabolizing slowly, in the dark, at temperatures a few degrees above freezing, at depths where no sunlight has ever reached and no surface ecosystem has ever exerted any influence whatsoever. That borehole is not exceptional. It is representative, Similar fluid systems have now been characterized in the Canadian Shield, in the Kapval Craton of South Africa, in the Pilbara Craton of Western Australia, in the ancient basement rocks of Scandinavia, Siberia, and Brazil. The pattern that emerges from their collective chemistry is not one of isolated anomalies. It is the signature of a planetary system, a global subsurface biosphere distributed through the crystalline basement of every continent, sustained by the same fundamental energy sources, populated by related microbial lineages, operating under the same thermodynamic constraints, running continuously for timescales that geological evidence suggests extend back to the early Archean, possibly to the origin of life itself. The word biosphere has always implied a sphere, a layer enclosing the Earth, bounded above by the limits of the atmosphere and below by the limits of the soil and shallow ocean sediments. That mental model, intuitive and geometrically clean, is wrong. The biosphere does not sit on the surface of the Earth, it penetrates it. It extends downward through kilometers of solid rock, following fluid pathways, exploiting chemical gradients, persisting in environments where the conventional criteria for habitability, sunlight, warmth, liquid surface water, are entirely absent. The biosphere is not a skin on the planet, it is a volume. Recalibrating that understanding has practical consequences that extend well beyond academic biology. The global carbon cycle, the planetary accounting system that tracks the movement of carbon between atmosphere, ocean, land surface, and geological reservoirs, has been modeled, refined, and debated for more than a century. Climate projections depend on it. Carbon sequestration strategies are built around it. Policy decisions affecting the energy systems of entire economies rest on the accuracy of its parameters. And until the last 2 decades, every version of that model was built without accounting for the biological activity of the deep subsurface. The methane produced by methanogenic archaea in marine sediments, the carbon fixed by chemolithotrophes in continental basement rock, the sulfur cycled by deep fracture communities over millions of years, none of it appeared in the standard equations. A variable of planetary significance had been missing from the most consequential scientific model humanity has ever constructed. The process of inserting it is ongoing. The Deep Carbon Observatory's decade of work produced the first systematic global estimates, but the uncertainties remain large. The spatial distribution of deep life is heterogeneous and incompletely sampled. The metabolic rates of deep subsurface communities are difficult to measure accurately. The standard radiotracer techniques used in surface microbiology require incubation conditions that are nearly impossible to replicate for organisms adapted to extreme pressure and temperature. New approaches, including in situ incubation systems deployed directly inside boreholes and geochemical flux measurements that infer biological activity from the rate of reactant consumption and product accumulation in fracture fluids, are producing more reliable data. Each new dataset narrows the uncertainty range. Each narrowing shifts the carbon cycle models. Those models matter for a reason that connects the deep Earth directly to the surface world in a feedback loop that is slow by human standards but geologically instantaneous. As ocean bottom water temperatures rise under current climate trajectories, the pressure-temperature stability field of methane hydrates, the ice-like structures in which microbially produced methane has accumulated over millions of years of deep biological activity. Contracts. Hydrates that are currently stable at their seafloor depths become marginally unstable. Methane is released into the water column and, in quantities dependent on ocean chemistry and depth, reaches the atmosphere. The additional atmospheric methane drives additional warming. The warming destabilizes more hydrates. The feedback is not unbounded. There are chemical and physical limits on how quickly seafloor methane can reach the atmosphere. But its magnitude and timescale are functions of the deep biosphere's productivity over geological time. The size of the methane reservoir that now sits in pressure-temperature stability at the seafloor was built by organisms that no climate model until recently knew existed. Deep life did not merely survive on Earth's margins. It participated in building the planet's chemistry. Sulfur cycling by deep subsurface communities over billions of years has influenced the oxidation state of the oceanic crust and the composition of hydrothermal fluids venting from mid-ocean ridges. Iron cycling by deep fracture organisms has affected the distribution of soluble ferrous iron in ancient groundwaters and contributed to the formation of banded iron formations. those ancient sedimentary rocks whose striking red and gray layers record the progressive oxygenation of Earth's early oceans. Carbon fixation in the deep subsurface has sequestered inorganic carbon into organic form in geological settings entirely disconnected from the photosynthetic carbon pump. These contributions are not large relative to the dominant surface biogeochemical fluxes at present. Over geological time, compounded across billions of years of continuous activity, they are not negligible. The astrobiological implications of all this have crystallized over the past 2 decades into a specific and testable scientific hypothesis. If life can survive and evolve independently of stellar radiation, fueled by rock chemistry and radioactive decay, in environments physically similar to the interiors of other rocky bodies in the solar system, then the question of whether life exists elsewhere in the solar system is no longer primarily a question about stellar habitable zones. It is a question about subsurface geochemistry. It is a question about whether liquid water contacts reactive rock at depth. It is a question about whether radiolysis or serpentinization produces hydrogen in quantities sufficient to sustain a minimal metabolic community. Those conditions are not rare in the solar system. They may be common. Mars lost its surface habitability approximately 3 to 4 billion years ago when its magnetic field collapsed, its atmosphere was stripped by solar wind, and its surface water disappeared into space and subsurface ice. But its interior retained geothermal heat longer than early models suggested. Liquid water may persist today in deep subsurface aquifers, particularly in regions of residual volcanic activity. The Martian crust contains iron and magnesium-rich rocks susceptible to serpentinization. Radiolytic hydrogen production from uranium and thorium in Martian basement rock is expected to occur at rates comparable to Earth. If life ever gained a foothold on Mars, and the evidence for ancient surface habitability strongly suggests the conditions for its origin were once present, the subsurface is where it would have retreated as the surface became inhospitable. The search for life on Mars, framed through the lens of the deep biosphere, is not a search for fossils of a dead biosphere. It is a search for a living one, operating at depths currently beyond the reach of any landed spacecraft. Enceladus is more immediate. The Cassini mission's direct sampling of its plumes, material ejected from the subsurface ocean through fractures in the south polar ice shell, detected molecular hydrogen at concentrations, indicating active hydrothermal reactions between liquid water and the rocky seafloor. The chemistry is serpentinization. The energy source is the same one that sustains Candidatus desulfuridus audax viator in South African basement rock. The organic compounds detected in the plume are consistent with hydrothermal synthesis. The pH of the subsurface ocean, inferred from plume chemistry, is alkaline, similar to the conditions proposed in the alkaline hydrothermal vent hypothesis for life's origin on Earth. Enceladus is a small moon, roughly 500 kilometers in diameter, orbiting Saturn at 1.2 billion kilometers from the Sun. It is, by every surface criterion, one of the least hospitable objects in the solar system. Its surface temperature is negative 200 degrees Celsius. It receives roughly 1/100th of the solar energy per unit area that Earth does. None of that matters. What matters is what is happening in the rock beneath its ocean. Europa's subsurface ocean contains an estimated 3 times the volume of water present in all of Earth's oceans. Its rocky seafloor is subject to tidal heating from Jupiter's immense gravitational field, a process that generates heat in the moon's interior through the continuous flexing of its rock, independent of solar energy and independent of radioactive decay. That heat drives hydrothermal circulation. Hydrothermal circulation creates chemical gradients. Chemical gradients are energy sources. On Earth, in analogous systems, those energy sources sustain life. The logical structure of the argument does not prove anything about Europa. It identifies a specific physical environment where the conditions that produce life on Earth are present, and asks whether the outcome is different. The answer to that question is not yet known. It may not be known for decades. The missions required to access Europa's ocean, drilling or melting through an ice shell estimated at 10 to 30 kilometers thick, represent engineering challenges at the frontier of current capability. The mission to return samples from Enceladus's plumes, which would require no drilling at all since the material is already being ejected into space, has been proposed but not yet funded at the level required for implementation. The constraints are not scientific. The science is clear about where to look and what to look for. The constraints are technological, financial, and political. What has already changed, permanently and irreversibly, is the conceptual framework within which those searches are conducted. Before the deep biosphere, the search for extraterrestrial life was organized around stars, around habitable zones, around surface liquid water, around planetary atmospheres detectable by remote spectroscopy. The discovery that life on Earth is predominantly subsurface, fueled by chemistry rather than light, persistent for geological timescales in isolation from the surface world, and potentially present throughout the rocky interior of a planet from its deep Archean past to its present. That discovery did not merely add a new category to the search, it relocated the search entirely. Life is not a surface phenomenon that occasionally tolerates extreme conditions. It is, on this planet, primarily a subsurface phenomenon that occasionally colonizes the surface. The surface biosphere, the one we live in, study, depend on, and until recently believed to be the totality of life, is the outlier. The rule, written in ancient rock and ancient water and ancient chemistry, is the dark, pressurized, radioactively fueled world beneath it. We have been living on the roof,